Hello, everyone, and welcome to the talk. I know I'm the last speaker of today's events, so I'll try to wrap up and give you guys a little bit of information about this more or less work in progress that my group in University of Chicago has been working on. And to just get everybody started on the same page, I think everybody here for the conference already are well known the power of vaccine to combat viral diseases. So here I'm showing you on the right-hand side the number of deaths caused by different viruses before and after the introductions of vaccines. And you can clearly see that the introductions of vaccines is our best workhorse to combat numerous infectious diseases, including smallpox, polio, and measles. And just to give everybody a recap of the different types of vaccines, we have these whole dead bug approaches, and these are the attenuated or inactivated vaccines. And there's also these sort of parts and pieces approaches, which usually instead of taking the whole bug, we want to use either their toxins or some of their molecular signatures or like a subunit of that pathogen to achieve that immunogenic response. And finally, there's more, I guess, novel approaches nowadays, which comes down to really the nucleic acid vaccines, so mRNA vaccines, and also virus-like particle vaccines. And in today's talk, I'll be focusing on our work on a virus-like particle vaccine, which we usually would just call them VLPs. And VLPs is really a vaccine based on the self-assembling nature of viral capsid proteins. So these viral capsids are able to form these subunits are able to self-assemble into either these trimer or pentamer shapes, and they can self-assemble into a larger either spherical or rod-like viral capsid. And unless I'm showing you guys a cartoon of how that looks like, and on the right-hand side, this is an electron microscopy image of actually how that viral capsid looks like. And you could clearly appreciate that the viral capsid is not sort of a uniformed mass. It's rather more so like a tile, more like a Lego pieces that are made out of individual subunits that makes up this beautiful structure. And what I'd like to emphasize today, though, is there has been a lot more reports on using virus-like particle vaccines rather than subunit vaccines, mainly because virus-like particle vaccines display these repetitive surface of antigens. So if we're comparing on the left-hand side, these soluble antigens, you can see that if you have your B cell receptors on the surface of your B cells, once your antigen binds to them, they're not able to cross-link between the B cell receptors. However, if you compare those antigens when they're decorated on top of your virus-like particle, that means many of the times your B cell receptors will be binding to the same VLP, and that causes a lot of cross-linking in these stalk regions of the receptors. And this is causing a very potent cross-linking effect that many people it's more like an empirical observation that people know for a long time that VLP are more immunogenic than subunits, but the exact mechanism of this is still being discovered and explored. And now the biggest issue with virus-like particle vaccine, though, is that as its name suggests, virus-like particle vaccines, you have to be a virus, right? But not all pathogens are viral. And today's talk, I've heard so many different talks about people working on parasites, protozoa, fungi, E. coli, bacteria, etc. So you could clearly see that if these pathogens are not viral in nature, how can we actually use virus-like particle vaccines on these sort of pathogens? So this comes down to the idea of a chimeric vaccine. So here I'm showing you sort of the basic idea is to have a multi-head and monstrous form of this mythology creature that have different heads from different organisms all attached to the same body. And this is exactly what chimeric vaccines are about, which is essentially instead of using viral-based pathogen antigens, you want to take your antigens from a non-viral organism and then somehow plug that in onto your viral-based particle to make this sort of chimeric VLP shape. And chimeric VLP, though, the biggest question that you might be thinking is how exactly we establish this linkage between this antigen and the base. So what we're looking at here is basically this line right here. How do we exactly establish that linkage? And numerous different attempts have been trying to figure out this exact line from different approaches. One of them is genetic fusion. So what you want to do is basically to fuse your antigen together with your scaffold protein by just removing that stop codon and adding a linker to it. But the problem with this, though, is that you're really taking a big leap of faith, because many times when you connect antigens with your scaffold protein, sometimes they'll misfold, and many of the times these scaffold protein are not able to self-assemble as properly as their wild-type counterparts. And another way of doing so is known as chemical cross-linking. So chemical cross-linking is you can install these chemical handles on both the antigen and your particle, and then upon either acid-base or redox reaction, you combine them together, and then this is how you sort of establish that linkage. But again, I like to reiterate, this process really takes a lot of time to formulate, because sometimes these self-assembling processes are very sensitive to salt concentration, pH levels. So many of the times, chemical cross-linking will cause a lot of these very distorted particles. So this comes down to a work by Cambridge. This PI known, his name is Mark Howarth, quite novel, quite well established in the world of synthetic biology. But it was back in 2012 where his group actually used a bacterial protein from S. pyogenes, and then this protein, they were able to split this protein into two parts. One they call as spike catcher, and the other part they call it spike tag. And the reason they're sort of like a catcher and tag is because immediately when you mix these two proteins together, they will able to spontaneously form this isopeptide bond again. So you can imagine that if your antigen has a spike tag and your particle has a spike catcher, then this is how you can establish that sort of linkage that kind of connects your antigen together with the particle without using any harsh chemicals. So we got really, really excited about this technology, and this paper, also by the same group, 4 years later, they actually used this exact technology for targeting malaria antigens. So what they did here is that they have the spike catcher, which is the spike catcher, which is the protein subunit that is able to bind to spike tag, fused with this AP205 construct. This construct is just the basic particle scaffold. So this creates this sort of like scaffold shape that they use as a presentation platform. And on top of that platform, they fuse spike tag with the malaria antigen. And this way, when they just mix two mixtures together, they're able to sort of create these plug-in and display decorated VLPs that uses essentially a viral base decorated with malaria antigens. They immunize, they immunize mainly with mouse models, and they showed a very strong, potent results that has at least tenfold boost of antibody IgG1 production, and they're still working on sort of pushing it further to clinical trials, but this is still work in progress. And inspired by their work, our team at UChicago kind of thought about how we can push this technology a little bit further. And one of the ways we thought this is very valuable is how modular this is, because once you have the viral capsid, you could basically plug in antigens from any pathogen that you can imagine. And you can clearly see that this is very valuable for many of the neglected tropical diseases, because many of these antigens are studied, but they're not yet in their most optimized form. So many of them are still in subunit form vaccines. So this is why we began to focus on this tropical disease known as schistosomiasis. And schistosomiasis is it's such a prevalent disease. It affects around 258 million people, and these are very conservative estimates. And schistosomiasis comes down to 3 different subtypes. One is Schistosoma mansoni, and then there's hematobium and also japonicum. And their availability is really limited to their geographic distributions. And I'd like to tell everybody a little bit more about how this parasite sort of matures, because it's a very interesting process. So essentially, these parasites rely on an intermediate host, a vector. These are the freshwater snails that harbor these parasites. And when these infectious parasite, when they're small, they're called Cercariae. These Cercariae shed off from these freshwater snails and then penetrate through typically people working freshwater. So these are farmers, these are women helping out with their household work, and these Cercariae will penetrate their skin where they go into the circulation and reaches their lungs. Eventually from the lungs, they will reach out to the heart, and then finally these Cercariae will mature and reaches juvenile phase in the liver. And eventually, the worms will become fully sexually mature in their hepatic veins. And this is where they start to lay a lot of the eggs. And the eggs are the biggest problem because the eggs are super immunogenic. And many of the times you see so much fibrosis in the liver tissue, causing a lot of pain in the patients. And finally, these eggs are excreted either through the urine or through feces, depending on the species of schistosoma you're talking about. And just to give you guys a little bit more about the geographic distribution that we're talking about here. So again, earlier on the previous slide, I told you guys there are three main species around the world, and one of them, Hematobium, Mansoni, and Japonicum. And you can see that Hematobium is mainly more so, it's more prevalent in parts of Africa, also in the Middle East. And Japonicum, however, is more prevalent in the Pacific region, and sometimes China has a very sophisticated network to provide surveillance for these Japonicum worms. But many times, whenever there's a flood, due to apparently it's a lot more common with global warming in the Yangtze River area, these Japonicum worms will still come back. So that's why this institution remains to be very stringent in their surveillance efforts. And finally, Mansoni is more prevalent in a lot of parts of Africa as well as Latin America as well. And these worms, their geographical distribution really depends on the availability of snails at that local region. So for example, this snail right here, which is the host for Hematobium, this guy is, these availability of freshwater snails directly determine where the parasites are forming. So here, I just shared how complex the schistosome life cycle is, right? So for example, they undergo two different host species, one stage as living in the freshwater snail, and then finally goes to the host. And also, there are a lot of drugs available nowadays. So the biggest workforce is praziquantel, but praziquantel has been administered in a lot of mass drug administration programs. But these the mass administration of these drugs actually cause a lot of praziquantel resistance, and also not all patients respond very positive to the treatment. So that's why we keep on pushing for a more long-term treatment or prevention method for this particular parasite. And also, when our group was sort of investigating more about this disease, we're so surprised to see that all of the sterile vaccines for this particular worm are in their subunit form. So what I'm talking about here is that here are the 4 existing vaccines for schistosoma, more for Mansoni, because Mansoni is the most well-studied strain. And you can see that SM14, this one is a fatty acid binding protein. Also SH28GST, this one is targeting S hematobium, and it's specifically targeting a S transferase in their metabolism. And another vaccine candidate is known as P80, and P80 is targeting the Mansoni strain's large subunit of the cowpain protein. And this particular vaccine candidate is currently tested with funded by the Gates Foundation, and they're still working on clinical trials right now. And another one is TSP2, which is targeting tetraspanin, and this is a small surface antigen that people actually found this out by computational screening, and this one is currently funded by the NIH to perform clinical studies. However, I like to emphasize all of these vaccines are currently in their subunit forms. And as I mentioned in the very beginning of the talk, we already know that subunit vaccines are not the most efficient in terms of reaching that immunogenicity. So what our idea of our group is to essentially use what Mark Howard did in Cambridge. So instead of using these just subunits, we want to design these antigens, express them together with spy tag, and also we want to plug them in onto our virus-like particle scaffold. So this way, your antigens are no longer just small little subunits, but rather this massive structure that your body can recognize as more immunogenic. And what's more exciting, though, is that a very recent paper from 2024 published in Nature, they actually did a massive screening on Schistosoma mansoni. And what they did here is that they have the whole cDNA sequence of Schistosoma mansoni, and they cloned them into a microarray, and then they allow these phages to express these subunits, and then each phase will express one single antigen. And then they just, at the same time, they also infected macaques and also hamsters with these infectious Saccharinae so that their sera contain a lot of antibodies. So when they mix them together, you can see exactly what antigen is bound to that antibody. And based on this computational screening, they were able to find a lot more candidates that people previously have never thought as antigen candidates before. And out of these, there's two very exciting ones that we chose to sort of focus on. This paper actually discovered more than 100 of them, but these two are just the more immunogenic ones. So one of them is SMAE. So this is the endopeptidase. This is their AlphaFold predictor structure. And another one is MEG 4.1. And MEG 4.1 is here, the graph right here is showing you when exactly after the infection that this particular protein shows a strong binding to the antibodies. And here you can see that couple of weeks post infection, and also you can see that clearly this enriched this enrichment of MEG 4.1 suggests that MEG 4.1 is another candidate gene that we can use for vaccine development. So here, just to reiterate, our whole mission is to both recycle these old antigens. So these are SM14, TSP2, and SH28 GST. So these have already been tested or are still going through clinical trials right now, and also to test these new antigens from phage display studies. So these will be SMAE and MEG 4.1. I'm putting a ... there is because based on how modular the system is, we can basically plug in whatever antigens of interest to accelerate that process. Again, we want to have these antigens together expressing the spy tag, and then we want to also have this scaffold protein, and finally conjugate them together to make this schistosoma antigen decorated virus-like particle. And what I like to just sort of sell this point a little bit more is that on the one hand, this vaccine platform has a lot of utility because schistosoma vaccines development has been such a slow process just because it is a very complex parasite, and there's been numerous efforts to do so, but we really want to use this modular, this sort of three-dimensional structure of the virus-like particle to enhance a stronger immune response and also reduce the doses and the use of booster to reach that optimal protection level. But also our platform also sort of offers this modularity in terms of accelerating testing, because you could plug in whatever antigen from multiple phases of the schistosome parasite life cycle. And also you could test different geographical strains, but it's very easy just to plug them onto the same scaffold. And we really hope that this platform can inspire other researchers to accelerate that vaccine development process. So I'm not going to go into a lot of the details of how you exactly characterize these proteins, but please take this as like a pioneering, like a work in progress that I'll tell you guys about. So to clarify that we have the right weight of the protein, we usually just run SDS page and also Western to confirm that it's right size after conjugation. We also purify them with size exclusion chromatography, and finally we ensure that our particles are the right size using using dynamic light scattering. Also, I like to keep in mind that schistosomes don't affect a lot of the developed countries. So this is it's very unfortunate that very many of the places that these vaccines have to go to will have to undergo very few sort of cold chain availability. So we also want to make sure our protein is robust enough so that if it is lyophilized, can they be shipped at room temperature? We also did lyophilization studies, assays, freeze-thaw them numerous times and make sure that our proteins are robust after we tortured them quite a bit. And finally, to understand whether or not the particle that we made are as immunogenic as we think, we are actually collaborating with a NIH-funded group. They're called Schistosomiasis Resource Center, and they are a specialized group that provides parasite life cycles to all of the different places of the world, and you could just order parasite from them, and then they will just ship it to you. It is a really awesome institution, and we've been collaborating with them. So we, as a university, we will ship them our produced proteins, and then they are currently testing the immunogenicity of these vaccines in mice. And what we plan on doing is really two phases. So first, that we want to vaccinate healthy mice and detect whether or not they actually produce an elevated level of antibodies. And however, this is clearly not enough because these are healthy mice, right? So we also want to go into phase 2, and we want to vaccinate these parasitized mice and see if vaccination actually provides protection against the worm damage. And based on this, I like to thank all of every single person who've been so supportive of the project. I am only representing my team as the project lead, but numerous people helped out on this project, and also our collaborator in Schistosomiasis Research Center. So I'll end my talk here and happy to take on any questions. Thank you.